Alright, welcome to our next video on genetic engineering, our third part of genetic technology. This is going to be where you could mix and match DNA. You can make a watermelon that's blue. You could make a fly with 8 wings. You could do whatever you like. You can mix and match genes from one species to another. Something that cannot naturally be done since two species cannot breed together. And there's some more applications. We'll take a look. But genetic engineering, pretty cool process, and any organism that's made using it will be considered a GMO, genetically modified organism. Let's take a look. So our problem here, we got people who are diabetic. They're getting insulin from cows and pigs, since pigs and cows are really genetically similar to humans. But there are some problems with this. The body's probably going to reject the insulin since it's not exactly human insulin and it's foreign. The white blood cells will recognize it and fight it off, make antibodies, and have a full-fledged allergic reaction. Also, there's not much insulin to be harvested. When you get it from a pig or cow, it's harder to get. And also, since we're really closely related to cows and pigs, there could be pathogens that could be transferred from cows or pigs to us that could affect us as well. So there's a lot of problems with using cow or pig insulin, which was used for a long time until genetic engineering took place. So the goal, we need to make insulin fast, safe, and in large quantities to bring down the price. How can we do this? Well, in order to start or understand this process, you really got to understand this thing called bacteria. And bacteria, if you look inside, is a circle. There's a bunch of circles and a squiggly line mess. The squiggly mess, that's messy DNA, referred to as chromatin. The circular DNA, really only present in bacteria, might be in viruses also, but generally bacteria. And this is very, very unique in that it loves to copy itself. It makes a whole bunch of copies. And this circular piece of DNA in bacteria is referred to as a plasmid. There's a special section on this plasmid, this circular piece of DNA in bacteria. Whatever gene is here, that gene will constantly be made. So you could put any gene you want in here, like an insulin gene or testosterone gene, and the bacteria is going to make that protein. So if you put estrogen in here, it's going to make a lot of estrogen. If you put growth hormone, this bacteria will make a lot of growth hormone. So this area of the plasmid, the circular DNA, we're going to cut it out using a restriction enzyme. It's called a restriction enzyme because it only cuts certain letter sequences or base sequences in DNA. So wherever it sees AATT, it will cut. Or if it sees CCGG, it will cut there. Depends on which restriction enzyme you're using. With that, we cut out that little section of DNA, whatever's there will get made. We cut that out, and we put the gene of our choice. It could be the insulin, testosterone, growth hormone, estrogen, any gene you like. We put that gene in there, and we make a thing called recombinant DNA. It's kind of like a hybrid piece of DNA. This piece of DNA, and you can see it still, it is right here. That's the recombinant DNA. That recombinant DNA is going to go into the bacteria. Bacteria love to absorb DNA, so they're going to take that right back in. Besides loving to take in DNA, they love to give it off. And bacteria also reproduce unimaginably fast. I believe the fastest bacteria they found to reproduce anywhere from 3 to 5 minutes. So that means in 3 to 5 minutes after bacteria give birth to a baby, their babies can make babies also. So boom. This bacteria is reproducing very fast, and it uses a special type of reproduction called mitosis. Mitosis makes identical copies. So every single one of these bacteria contain the gene for, let's say this is insulin. Every one of these has the insulin gene inside it. So all of these bacteria will produce insulin. And they produce it very quickly. And if you have a lot of something, the price of it will go down. Supply and demand. So this insulin in this vial, it's going to be cheaper. Besides being cheaper, it's also going to be safer to use because it's going to be made using the insulin gene from humans. So it's literally the instructions to make human-specific insulin. So it's safer, it's cheaper, and chances of getting a pathogen, very low because this is pure insulin in here. This is the first way genetic engineering or gene splicing, bioengineering, whichever term you like to use, this is the first way it could be used. But there is another way this technology can be used. So this technology can be used to make genetically modified organisms. A problem here, there are insects or pests eating farmers' crops, spraying super costly, very time consuming, and it kills everything, even the good insects like the bees. So our goal is to make a genetically modified organism that makes its own poison, so the bugs that go up to the plant and bite it, they'll die, and the good ones will live. Let's take a look and see how can we make a plant that will kill only the bad bugs. We need to look at our bacteria. We need to remove the plasmid, that circular piece of DNA, and we're going to cut it up. In order to cut DNA, you need something very specific, a restriction enzyme, restricted to only cut certain letters. So we cut out that special section of the plasmid, and we're going to insert our gene of choice, a poison gene. This gene kills insects, or makes a protein that kills insects. We're going to add this gene to the bacterial plasmid. And we make a piece of DNA called recombinant DNA that we saw before. Kind of like that hybrid DNA, two different pieces of DNA from two different species put together. This recombinant DNA is going to be absorbed by bacteria because they love to take in DNA. And bacteria divide super fast by mitosis, making identical copies. Now we're going to get a plant cell from this corn crop that's being killed by insects. And we're going to use enzymes to break down and digest that really tough cell wall made of a material called cellulose. So the enzyme that will digest it, you guessed it, cellulase. And now we have a plant cell that's naked, so to speak. There's no cell wall, just that plasma membrane or cell membrane. You put that genetically modified bacteria next to or with the plant cell, and the bacteria will give the DNA, or will give that specific gene to this plant cell. And you can see the little red gene, that poison gene, it is now part of the plant cell's genome, or part of the plant cell's DNA. This plant cell now has the capacity to make this protein that's a poison, and it kills specifically insects, because insects have specific cell receptors that are different from ours, so we won't be affected by this poison, but the insects will. This one cell that makes the body of the plant has to divide like crazy to make a full plant. So body cells divide by mitosis. and it's going to divide into a ball of identical cells, all with this poison gene, and it's going to make eventually a big plant that will go in the ground. And since all the cells of the plant body were made by mitosis, all those cells in the plant had the poison, or can make the poison, I'm sorry. If the bug eats any part of that plant, a leaf, a stem, it will die because all the cells make the poison, because they all came from that recombinant or genetically modified cell. The bad insects will die, the good insects are not affected. This is now considered a GMO, or genetically modified plant that makes genetically modified food. So here's a little extra story. Small nation struggling to feed its people. Crops are having trouble growing near the salty waters. Call a biotech company. They say, hey man, you guys got to insert a salt-resistant gene into our plant's DNA. They have to be able to survive the salt water. Scientists introduce the salt-resistant gene, and they can now be resistant to salt water. Big positives for this technology. You can make human proteins, or meds, fancy word for medicine, like insulin. Human insulin, that's a medicine for diabetics. You can make them fast and cheap. Remember, if you make a lot of something, the price goes down. Supply and demand. Also, they're going to be much safer since you're using human-specific genes, like the human-specific insulin gene. Less likely to be rejected since it's specifically made for humans. The white blood cells won't recognize anything as antigens. Also, you can combine, or you could add, desired genes and thus traits in organisms to make them the way you like. Those will be called genetically modified organisms, or GMOs. Bunch of negatives though. So if you change one area of DNA, there's a high chance that you could inadvertently or mistakenly change another area of DNA, changing a gene, causing new proteins to be made in plants and foods that we eat. That could cause people's bodies to have an allergic reaction to these new proteins in foods like peanuts or shellfish, leading to more allergies. And GMO plants, they could also create resistant insects, because only the insects that bite the plants will die. But some of them that bite the plant won't die because they're resistant. And those bugs that are resistant to the poison, they'll live long enough, pass on their resistance, and you'll have a large population of resistant bacteria, or resistant pests, I'm sorry. That's a little segue into evolution. Little story here. Farmer wants to make blueberry plants that are large and sweet, and he has a large unsweet one and a small sweet one. How can he make a combination of large and sweet blueberries? Take the sweet gene and you just simply cut and paste it into the large blueberry plant's DNA with the help of bacteria. This genetically modified blueberry plant will now produce not only large blueberries, but sweet. You'll have a hybrid plant, or a genetically modified blueberry plant. This process, you guys know now, is genetic engineering, gene splicing, also called bioengineering. But there's a second way you could achieve this. You could selectively breed these plants. Take the large, unsweet blueberry plant and pollinate it with the small, sweet blueberry plant. And the babies you get will be large and sweet. Not guaranteed, you'll still get some variation, but if you do get the correct combination, the correct hybrid, you just do inbreeding. Take all the plants that have the combination you want and you mate them together. But again, it's not guaranteed because sexual reproduction does lead to variation. But on the other hand, genetic engineering, this is guaranteed. You're physically putting in the genes that you want, so you know the organism will have the genes that you want. And that is genetic engineering and gene splicing.